Hi, everyone, and welcome to the Cancer Research UK Cambridge Centre podcast. My name is Hui-Ling, and I'll be your host for today's Ask Me Anything episode. Hope you enjoyed. Today, I have with me three experts working around cancer-associated cachexia to talk about the science and challenges of detecting and treating cachexia, and what are the strategies for directing the future studies. Before we start, let's have our guests introduce themselves. So I will start from Claire, please.
Hi, my name's Claire Connell. I'm a clinical lecturer in medical oncology, splitting my time partly working in our oncology clinics, and part of my time is spent working within our research laboratory here at the CRUK Cambridge Institute. And I'm working on a clinical study aiming to better understand the underlying cause of cachexia in our patients.
Thank you. And David?
Hello, I'm David Lewis. I'm a professor of molecular imaging at the University of Glasgow and also at the Cancer Research UK Scotland Institute, which is the sister institute to the Cambridge Institute that Claire works at. I'm also the co-director of the Scotland Total Body PET facility, and my research in cachexia is really looking at molecular imaging technologies and how we can use those to identify cachexic patients early and some of the underlying factors.
You and Tobias?
Hello, my name's Tobias Janowitz. I'm a medical oncologist who used to work and live in Cambridge in the United Kingdom. I've recently moved to Cold Spring Harbor, which is a research facility close to New York City. And there I lead a research program focused on the mechanisms and consequences of cancer-associated cachexia.
Thank you very much. So let's start from our first question. So can you give our listeners a brief overview of what cachexia is, please?
Yes. Now, we know that patients with cancer can be affected directly by the cancer itself, but they can also be affected by the cancer exerting effects on other parts of their body. And cachexia is the process by which cancer affects multiple parts of the body to result in loss of appetite and loss of weight, with a loss of muscle and fat stores. And what we find is that these patients often suffer from significant fatigue and a decline in their physical abilities, as well as a general feeling of not wanting to take part in activities that they may previously have enjoyed. We also know that cachexia can reduce the ability of our patients to manage with treatments and, crucially, to benefit from these treatments. Now, cachexia is very common in patients with cancer. Most commonly, it's affecting patients with pancreatic, lung, and bowel cancers, but it can affect people with other types of cancers. And it's a real challenge because it causes suffering of patients and their relatives, and yet we have no good treatments for it.
And would you like to add anything, Tobias?
No, I think that's a very good summary. I think it is important to realize that cachexia is not particularly well understood at this point. So we have, as clinicians, this diagnostic term where we essentially capture patients who are in themselves unwell as a consequence of cancer, and that is mostly defined through involuntary weight loss. So in other words, the weight drops off, they lose muscle mass, they become fatigued, even though they would prefer to be better.
Thank you. Following the first questions, could you specify again what are the primary symptoms and signs of cachexia? And also, Tobias, you mentioned about the weight loss. So is there any other different type of weight loss condition and how this would differ from cachexia?
Yeah, thank you. I would like to remind the audience perhaps where this term came from. It was first coined by Greek physicians who called it cachexia, which is from kakos hexos, which means bad condition or feeling unwell in the Greek translation. And really the primary symptoms and signs refer to this. Patients often come to the clinic and ask very good questions. They ask, for example, if I'm losing so much weight, why am I not hungry? And this sort of seeming paradox of weight loss coupled with loss of appetite is probably the defining diagnostic sign of cachexia. When Claire and I see patients in the clinic and somebody comes to us and says, I don't know what's going on, I'm losing weight, but I'm not hungry, we would consider that a very significant statement because it would tell us that they may have cancer or that their cancer might be progressing. I think, what is the difference to other weight loss conditions? Think, for example, about a patient who may have cancer and the cancer blocks their digestive system. Say, for example, the food pipe that they use to swallow the food from the mouth into the stomach. It's called the esophagus. That might be a mechanical obstruction. So in other words, they may truly physically struggle to swallow the food that they've eaten. And as a consequence, they may well lose weight. But they will be hungry, and we can treat that condition successfully by bypassing the obstruction and feeding the patient. There's a clear distinction there between weight loss secondary to the inability to eat and weight loss secondary to the condition of systemic weight loss and muscle loss and fat loss coupled with loss of appetite and interest in food.
I don't know if it's helpful, but I just wanted to have had a quick question. What, how often, say for example, pancreatic cancer, is weight loss the primary presenting symptom? In other words, as you kind of pointed out there, Tobias, people are coming, they might not know they've got cancer. So is it a large percentage then of people who come with finding that they're wasted and then they...
You know, David, that's a great question. And that will actually come right into your forte in a minute when we speak about imaging technique, because I think that has changed over the last maybe decade or so. It used to be the case that patients regularly would come to the pancreatic cancer clinic and had been picked up in the referral pathway by their GPs because they presented with involuntary weight loss and wasting, and the GP would be worried. And that would really be the presenting symptom and sign. And then they would get workup. I think over the last decades, this has shifted more as imaging techniques and screening techniques become more widespread. There is now a fair share of patients who get referred to the clinic because an area in their pancreas has been identified during an imaging procedure that they may have received for a completely different reason that looks suspicious, and then they get worked up for that. But those patients, even when they get diagnosed early, very frequently in later stages of the disease, will still experience cachexia because the cancer frequently progresses. Pancreatic cancer, for example, is very difficult to treat. And therefore, cachexia becomes, again, a universal problem that ultimately affects everybody, if not at the presentation of the disease.
So it's interesting, and the early detection is actually feeding into, advanced detection of cachexia as well, but just early detection of cancer, which is obviously growing a lot as a specific area.
Correct. And I think we should speak about that more when we talk about the research avenues.
Yeah.
And I just want to add another follow-up question because, Tobias, you mentioned that for the patients, they're going to the clinic and they just describe they are losing weight. However, they don't feel hungry. So does that mean, is there any kind of correlation between your brains, which kind of managing the digestion or feeling hungry, thirsty, these central nerve system with the condition of cachexia?
That's a very insightful question, and that is exactly what is happening. So our body has developed many systems by which it can sense what is happening in the organ system of the body, and it senses it in the nervous system and in the brain, and then uses that knowledge to drive behavioral changes. And we are increasing the understanding that that is one of the causes of cachexia, that tumors release factors or signals that can be sensed by the brain, and the brain then structures the behavior that it directs in a way that patients and or model systems that we study in the laboratory display these behavioral changes that we associate with the sickness behavior of cachexia.
And I believe you have already touched upon about some effective treatment for cachexia. And would you please, specify, is there any other interventions that currently clinical use for cachexia?
Perhaps I make a start on this because I want to follow up from the causality question that you raised before. Medicine these days develops in a way that we try to understand the molecular process that drives a disease, and then we try to target that molecular process to improve the condition of the patient who suffers from the disease. And cachexia is no different. So we are currently trying to understand processes for which we know the molecular switches that might drive cachexia, and then we target them. But we are not quite there yet. And by that, I mean, if you were to ask me what is a treatment for cachexia, I would have to say to you, we do not currently have a good treatment for cachexia, and we do not currently have a clear target for treating cachexia. There are, however, very, very good hypotheses and ideas coming through, and we are beginning to understand some of the defining mechanisms of cachexia so that we can hopefully target them. And I think we are only a few years out from seeing some true interventions that will help some specific patients who have cachexia. Claire, maybe you can comment a little bit on the supportive care that we currently offer to patients with cachexia.
Yes, thank you, Tobias. So within the NHS, when we're seeing patients with cancer cachexia in our clinics, we think about ways to improve their nutritional status. We work closely with our dietitian colleagues to give advice about how to increase the nutrients they're eating. And we also think about potential reversible causes of their reduced appetite and their weight loss. For example, we know that a small amount of nausea can take away a patient's appetite. So we work to keep nausea well controlled. We can also think about how taste may be affected. For example, we'll look for fungal infections within the mouth called thrush, and that can cause a white coating to the tongue and is very common in our patients. It leaves the mouth feeling very dry and takes away the normal taste of food. So we'll aim to treat this to restore the taste of food. We can also think about whether the patients are able to absorb the nutrients in the food properly. For example, if we find they are deficient in some of the digestive enzymes, then we can replace these. So it's all about trying to treat any reversible causes of the weight loss and to increase the amount of nutrients their bodies are receiving.
Thank you. So starting from here, we will now shift our focus from the experiences of doctors and patients in clinic to the groundbreaking research happening in the laboratory. To better understand the biology of cachexia, researchers have already conducted some preclinical studies using mouse models. These findings will still require further clinical validation. So first of all, Tobias, can you share any recent advancement in studies of the relationship between the diet, cancer, and cachexia, please?
Yes, thank you, Hui-Ling. I want to first emphasize what you've just said. It's really important at this stage to mark that we are now transiting to discussing research studies that are mostly focusing on model systems in the laboratory. And here we use mouse models of cancer and cancer cachexia. It's worth noting that the mouse has been a very good model system for cancer and for cancer treatment. And we have robust systems where mice with cancer develop the symptoms and signs, as far as we can assess it, that patients with cachexia have, in particular the weight loss and the loss of appetite. So we have some confidence that the mechanisms that we study in the mice might be relevant to humans. And we make efforts to confirm this with the studies that Claire, for example, will discuss later on, to validate in humans what we find. And we've made several discoveries in that area where we looked at nutrient intake, so food intake, and nutrient processing. So how is the food used in the organism and in the tumor to fuel the tumor growth or the maintenance of the organism? And we've made two important discoveries. One is a small molecule called interleukin-6, which is associated with inflammation and can be released by the tumor, can change in the brain how the drive for taking in nutrients is communicated. So in other words, it can drive in model systems a lack of interest in food in a certain way, and therefore drive an interest, a lack of nutrient intake. And it can change how the peripheral organs process the food. And that's important because cancers need nutrition and the organism need nutrition. And as we understand this better, we might be able to target the cancer without impairing the organism. And we are very sensitized to that notion because when we tried a nutritional challenge for the cancer, we found that that was also a very strong nutritional challenge to the organism. So instead of getting a better outcome from cachexia, whilst the tumors were smaller, we got a worse outcome. And so we had to counteract the effect on the host organism to get a better effect against the cancer. And I think this illustrates this interplay between the tumor and the organism that has the tumor. And now that we understand it better, we might be able to deploy diet in combination with anti-cancer treatments to open up the therapeutic window where we can push back against the cancer without pushing back against the host organism. So keeping the organism well whilst we treat the cancer.
Thank you. And another add-on questions, because you mentioned in the first part of your answer, the inflammation and cachexia, and also you mentioned about interleukin-6. So could you explain to our listeners what interleukin-6 usually do, and also whether this is like physical or pathological roles of these kind of like inflammatory factor, please?
Yeah, those are very, very good questions, Hui-Ling, and I wonder if you should be an advisor to our laboratory and to our research program, because if you think about maybe the principal observation in cachexia, you would lose weight if you have a negative energy balance. In other words, you use more energy than you take in. And you can perhaps think about why you would lose more energy than you take in, because you eat less, or you burn more, or you break down tissue that you need to maintain at the expense of eating. And so cachexia falls broadly into those three categories. We now know that there are some hormones released that mostly work through reducing food intake. Then there are some factors that seem to change both food intake and food processing. And then there seem to be some factors associated with cachexia that target mostly the fat tissue and the muscle tissue and drive their breakdown. And interleukin-6 probably falls into that middle category. It has an effect on food intake and on food processing. And I want to come back to your question where you said, what is the biology of this? Is this pathology or is this biology? And the truth of the matter is we don't quite know, but it is interesting to observe that cachexia does not only occur in cancer. It occurs in many other inflammatory conditions. And if you ever had a viral infection, and maybe the listeners can relate to that, when you had a bad flu, you go a little bit through a mini cachexia because you lose your appetite, you lose your energy, you withdraw a little bit from the world that surrounds you. You're not so interested in meeting your friends and doing the things that you normally enjoy. But then you recover and then you get back to your normal self. And patients with cancer feel like that, but they don't recover because the cancer stays. And now imagine feeling like you felt when you had the flu, but feeling like that all day, every day. And that's what patients with cachexia describe.
Then my next question is, how does your observation in the laboratory would associate with the current clinical practice?
Can I just, it wasn't not exactly a follow-up statement, but it was more just a kind of follow-up conversation point from what Tobias was saying. When it comes to the mechanisms and causes of cachexia, Tobias, what I think is fascinating to me is, and you're saying there's these factors which predominate in these conditions, which are leading to reduced food intake, but they would get so detrimental as to risk the organism when obviously fundamentally there's so many processes in place to stop that happening, because it's so important for survival of every organism on the planet that they don't waste away and die. And yet, and we know there's a lot of redundancy in those systems, because that's why obesity is so hard to treat, because you push the system one way and it pushes back. It's fascinating to me how that system therefore breaks down in cachexia, and obviously there's these molecular mechanisms underpinning them, and there's these antagonistic functions, but I mean, something fundamentally breaks, and it's very counter to evolution that, the host would be sacrificed in that sense. I just wondered what your thoughts were on like the sort of critical imbalance of these homeostatic mechanisms, which clearly occurs.
I think that's a fascinating question, and that's part of the current research program that we are all involved in, to try and understand to what degree is biology and pathology connected or intersected here. In other words, to what degree is this normal and sustained, or to what degree is this a broken normal and should be corrected? I think there are a number of processes that are known in biology where something is ultimately non-sustainable. And so I wonder if cachexia falls into that category. And maybe I don't have quite as much a view that it is broken necessarily as you do. I think it could be adaptive, but non-sustainable. Say, for example, if you wanted to run a long distance at a high speed that you can't sustain, it either becomes, as you say, either your body tunes you down, but if there is no way to tune you down because the underlying drive is not there, you will ultimately not be able to sustain it and have a fall or be exhausted, deeply exhausted. And I think cachexia might fall into that category. But I think I agree with you. It's a fascinating condition, and a lot has to be done to understand it better.
Yeah, it gets to the kind of core of survival of, the species in a sense. And that's why it seems so counterintuitive that this would predominate. I guess it's a bit like the kind of selfish gene. There's something that's more important than the individual biologically going on there, which is really interesting. And I mean, your analogy about the exercise, I suppose, and anybody who's tried to sustain a run for a long time knows that you can't do it to death, even if you wanted to, because you do run out of steam. But yeah, it's an interesting point.
Let me talk to Hui-Ling a little bit about this question that you just asked, Hui-Ling. How does our observation associate with the clinical practice? I think that's a great gateway into the sort of areas of expertise of Claire and David as well, in that I think it's important to highlight that we are currently at the stage of trying to transplant our knowledge from the laboratory into the clinical setting and test. Is it relevant? Does it hold up? Can we modulate some of the processes that we are understanding from preclinical work? And that's not only related to our own research, that's related to the entirety of the field. because we are, of course, not the only people who work on cachexia. And so we are currently in this process as a field and also as a group specifically in my lab as well, to find out if what we've discovered in the lab holds up in the clinic. And we do that usually in several stages. One is we ask for patients with cancer to become part of clinical research studies where we try to understand what is happening to their body in more detail. And we then, when we have an idea of what we can treat, ask them to be part of clinical trials where we try to target what we think is relevant to see if we can improve their condition and their life expectancy and their experience as they are going through cancer treatment. And importantly, if we can improve their ability to receive anti-cancer treatment. Because you have to remember, many of the anti-cancer treatments are also a challenge to the organism. And so if you keep the organism well, you can treat patients perhaps better with anti-cancer treatments. And so we are currently at this stage, and this stage, and that's really relevant, I think, to your listeners as well, is a stage which is a joint effort between the researchers, the clinicians, and the patients and their relatives, because we all make contributions to it together. And we are very grateful always when patients engage with us in those stages, ask questions that drive our research, but also participate in studies that we conceive to validate what we found in the laboratory.
Thank you very much. And now I would like to direct the question more toward the detection part of it. So this would be for David. And could you name, are there any diagnostic tools so far for detecting cachexia early? And also, why is early detection crucial for such effective treatment?
Yes, thank you for the question. As we've discussed already, one of the issues with early detection of cachexia is early detection of cancer, which is still an emerging important area. So, often patients presenting with weight loss as a symptom means that we're not detecting cachexia early, even though it's something that we associate with advanced disease. So there's something implicit in that about how we manage early detection of cancer that we need to address. But I think, again, critically, one of the other major issues with early detection of cancer is again, our lack of understanding of the molecular pathways that we need to identify the underlying causes of cachexia, because those would give us clues to what would be the right biomarkers. I'll defer to my clinical colleagues Claire and Tobias on how some of these tools are typically implemented in the clinic, but largely it's done as a sort of multimodal strategy to look at, for example, nutrition, physical examination, some blood tests, and more latterly imaging studies, and I'll talk about those more in a minute. But essentially, there's no single definitive test for cachexia at the moment. That's the bottom line, and actually, because some of the diagnostic criteria aren't completely clear cut, then it can be a progressive diagnosis as well. And fundamentally, that this lack of early detection is very hard to manage. So reversing weight loss in cachexia doesn't work particularly well, and so we anticipate, but we don't get much opportunity to prevent weight loss before it happens, even though in those cases as well, it's highly likely that these patients are going to go on to deteriorate further. But it gives us the best chance for new treatments coming online that we can hope to maintain weight in specific patients. I don't know if Tobias and Claire want to comment on the diagnostic tools from a real life clinical perspective.
Yes, as David has mentioned, accurate detection of cachexia in the clinic is currently limited. We mainly base it on reported weight loss and loss of appetite. However, our patients are often presenting unwell and may, for example, have an accumulation of fluid within their abdomen, and this can mask weight loss. And so having an accurate marker of cachexia that would enable early detection, or even better, an increased susceptibility to the subsequent development of cachexia, would be very helpful.
I'd like to say, it's always important to highlight that the imaging technology has come a long way, and radiology in the clinic is also very important. There are really clever things that David does in the lab that are not yet available in the clinic, but principally the insights that we get from imaging have been good, and people have made a significant progress, for example, in reconstituting the composition of the body from CT scans. Many of your listeners will have come across these scans where patients are put into a tube and then get scanned with X-ray quickly back and forth. It's called computer tomography. And while we mostly use that technique to monitor the cancer, we actually get additional information from this technique. And people like David have tools in their lab where they can calculate out what part of the body is muscle mass, what part of the body is fat mass, what part of the body has been replaced by tumor. And then we can look at these things combinatorially to understand better where is a patient, because you have to remember, we get those sets of informations repeatedly on the same patient. And that's a big, big power of imaging. So you can actually track a patient over time very closely, because you can do a computer tomography more frequently than you can do, for example, a biopsy or take tissue samples. And that's really an important set of tools to inform both the diagnosis, but ultimately also the monitoring of treatment success.
Yeah, the big advance there has really been in the computing power to process these images. Obviously CT's been around for a while, and while the quality of the CT scans is improving, essentially it's still doing the same thing it's always done. And these images are sitting in repositories, and there's not enough radiologists in the world to analyze these quantitatively, but really new tools using things like artificial intelligence, deep neural networks, the same sort of things that run things like ChatGPT, which you might be more familiar with. These things now can segment, hundreds of tissues within the body, really to get a detailed map, as Tobias says, and that's very accurate now, happens much more. And it's really computer-assisted radiology, which has really driven a lot of those advances. And those things are really helpful also in the case of early detection for things like mammography and other areas. So there's been a huge increase in that, and it's continuing. And I think some of those tools and technologies are still developing and experimental, and we would expect very soon, and even now, some of those coming into routine clinical practice.
And so, Devin, you mentioned about this non-invasive imaging techniques. Could you elaborate more of your work on this?
Yes, happy to. Yeah, so as Tobias mentioned, computer tomography effectively makes a 3D map of the body based on X-ray, serial X-ray. So a spiral X-ray can build up a total picture of body composition, but it principally looks at tissue density, and so it really builds a structural map. So you can identify differences between soft tissues, bones, organs, etc., quite exquisitely. What it can't do, though, is understand really functionally what's going on in those tissues, and so it can't look at the molecules within those tissues. And so we develop other technologies. One in particular that we're developing is called positron emission tomography. So these are PET scans. They're routinely used in the hospital currently to look at primary cancer diagnosis and looking at staging of disease, so whether cancers have spread, and that's really important information in terms of managing patients, whether they should get surgery or in the case of not getting surgery. So this technology is very sensitive. we can we label molecules, nutrients, and we can track those in the whole body over time. So typically, in the case of PET scans that you might get in the hospital currently, that is a radioactive sugar. A sugar analog, which then we can look where that sugar is going. Tumors are very hungry. They're growing all the time, and so they're very energetic, and so they take up a lot of this sugar, and that helps us identify them. But as Tobias was alluding to earlier, the sugar is only one nutrient. And what we're really interested in now in terms of cancer, but also in this context of the whole organism, is what other nutrients are at play, and how are these other nutrient systems interacting in the whole body, that might predispose to cachexia, that might give us some clues about the primary cancer. And so work in my lab is really building up a pipeline of tools, of molecules, of nutrients, things like sugars, fats, proteins, amino acids, labeling those and tracking those in the body over time. We can inject patients and look to the uptake of these particular nutrients in the tumor, but also in these other organs. So current work looking at things like dysregulation in the liver, we can then see that uptake of different nutrients in those organs and how they're redistributing. And importantly, we can do those measurements repeatedly over time, and we can look at how these things are changing. So we're hoping that some of those things will give us key insights into early detection. So some of those molecular pathways are changing early in cachexia, but it also gives an idea of what different kinds of cachexia patients might be getting. So what are the different mechanisms underplaying? And if we can identify those early enough, we can then start to put those patients onto specific treatment pathways.
Well, that sounds very promising, non-invasive tools for detecting cachexia. And out of curiosity, there's other radiation-based detection approach. For example, the low-dose CT, which has been well implemented in terms of early detection of lung cancer. So when it comes to the radiance exposure, could you suggest how much is in the PET scan that you're developing compared to, for example, the low-dose CT?
Yeah, so PET scans typically use more radiation than a low-dose CT. It's more akin to probably a diagnostic CT scan, so a standard CT scan, and that has limited a little bit the use of longitudinal PET scans in cancer diagnosis and in other conditions. We and others have developed or are implementing newer technology in PET now that uses much less radiation. So these are total body PET scanners that fit the entire patient in. And so rather than scanning the patient in small sections, you can capture all the radioactivity in a patient throughout the whole scan, and the radiation dose from those can be much lower. So actually similar to Tobias is in New York, and so when we fly over there, we get some radiation on the plane, and so we can get doses down to much more levels that you would experience in kind of day-to-day life, and that really changes our ability to do early detection with these technologies, because you can look at larger patient cohorts and you can look at them much more repeatedly. So I think that's an advance that will really improve the applicability of PET in many areas.
Thanks, David, for the information. Here, I would like to emphasize again that currently, researchers are looking at the advanced imaging technologies as part of selected research studies with very small number of patients in order to understand what the potential utility is and what information can be derived from them and how they can help researchers to develop further treatments. Now, I'd like to move on to a clinical study named MyPack. So, Claire, can you tell us about the MyPack study and what is this study aiming for?
Yes, certainly. The name MyPack stands for Metabolic and Immune Profiling in Patients with Cancer, and it's funded It's a study funded by Pancreatic Cancer UK. The MyPac study was set up because our ability to generate effective treatments for cachexia in humans is limited by a lack of thorough understanding about its underlying cause. We also do not fully understand the effect that cachexia has on the immune system and how this may be affecting treatment outcomes. And so the MyPac study aims to bring together scientists and doctors with expertise in cancer, metabolism, and the immune system to better understand the underlying cause of cachexia in humans and its effect on immune function. And we hope that this will really complement the laboratory work that Tobias has already touched upon. Now, the study set up so that our participants come in for an overnight stay on our research facilities, and we perform detailed analyses to investigate how cachexia changes aspects such as how we sense hunger and the drive to eat, how it changes the way we metabolize the nutrients in food, and how it affects the amount of energy our bodies are expending. And I think important to note, this would be a good opportunity just to highlight how grateful we are to all of our participants who have kindly taken part in the study so far, as we really appreciate that the study takes up their precious time, and often during a very difficult period in their lives when they may not be feeling well.
And the next question would be what would you anticipate from the MyPac study?
We hope that the MyPac study will improve our understanding of the underlying cause of cachexia. The aim is that we will facilitate future research to identify potential strategies to treat cachexia, and also to identify ways to diagnose cachexia earlier to enable earlier treatments, as David has already alluded to. From the immune perspective, we hope that the study may shed some light on why patients with cachexia tend to have reduced responses to treatment, and to facilitate future research to overcome this.
So for all of you, what challenges do you or you think that in general the researchers faced when they are studying and developing treatment for cachexia? Maybe I will start with Tobias.
Thank you. I think one of the biggest opportunities within cancer cachexia research, as you gathered even from interviewing the three of us, is to bring together synergistic expertise to take a fresh look at the problem. And I think you can look at this as a challenge, but I'd like to think of it as an opportunity, because we have built teams now of basic scientists who study molecular processes within the cancer cells and the tumor microenvironment, with basic scientists who understand inter-organ communication, with clinician scientists and clinical researchers who understand how these processes can be studied in humans and how we can study interventions to change these processes in humans. And then people like David who help us think about across species, how can we track and understand these processes better over time using sophisticated methods of monitoring and targeting and challenging the processes that we think are relevant to cachexia. So I think what we really have as a challenge is to try and convert this to a meaningful treatment for a group of patients with cachexia. And if I was to sort of formulate one real challenge to the field, that should be it. In the next 3 to 5 years, I think we should be able to demonstrate as a field that this effort can actually lead to an intervention that makes a difference to some of the patients who are at risk of developing cachexia or who have cachexia.
Then, David, do you have any other things you want to add?
Yeah, I think I agree with Tobias, and just to highlight, of course, we know all our patients are different and their cancers are very different. We still don't really know whether we'll be able to find one underlying common cause of cachexia, or whether there are different causes in different patient populations. So hopefully the research that's currently being undertaken will provide some insights into this.
Yeah, I mean, I agree with Claire and Tobias there about the real challenges and opportunities of getting our basic science into real working diagnostics and what we can achieve. I'm hoping that as an addition to looking at new targets and new drugs, we will also see an opportunities for drugs that are already existing to be used in the context of cachexia, because a lot of those targets might not be understood well in cachexia. The challenge there is, I think, trying to get that into large patient populations without necessarily being able to do big phase 3 trials can be difficult when you are repurposing drugs. I'd be interested to know what the Tobias and Claire think about that. How we take potentially small trials with promising information and put those into much more widespread practice. That can be difficult with, for example, non-standard interventions such as supportive treatment or combination treatments.
Yeah, I think that's probably a question that goes way beyond cachexia, in that there's a universal question: how do we develop treatments in medicine? I think what is part of this answer is, and that sort of goes back to the initiation of this interview, is what is cachexia? Once we understand better what cachexia is specifically for patients or for groups of patients, then it will be much better. Then we will be in a much better position to target that specific process that affects that group of patients. And ultimately, we have to find ways to develop therapies effectively across the board. That doesn't only affect cachexia. At the moment, one could argue that clinical trials are too costly to test the multiplicity of potential treatments. I think it is interesting, since you were asking, Hui-Ling, about the challenges. There are challenges that are biological and scientific, and then there are challenges that are more operational. And one of the challenges that we have, I think, even though within our community now as cachexia researchers, we all understand that cachexia is an important problem, I think we still have to bring that message out stronger to the community of physicians. We have to empower patients to understand what the condition is that they are having, so that they can ask the physicians, do I have cachexia? What should I be doing about it? And really bring out this information challenge and address it. And then the last bit, and that's related to what the point that David just made, is there's always the challenge of obtaining funding to ask them and answer the most important questions. And if we all agree, and if there's more consensus in the larger community that cachexia is an important problem, then one of the biggest challenge for us as a community is to secure the support from those who give funding to research in cachexia, so that we can continue the work that we have started, hopefully to ultimately help patients better.
Yeah, so I think Tobias has already touched upon that, about the funding opportunities that could help facilitate future research in cachexia. So for all of you, I believe that you all are belonging to the CANCAN team for the Cancer Grant Challenge. In your opinion, how do you think these Cancer Grant Challenge could facilitate tackling of these challenges in studying cachexia?
Yeah, maybe I can make a start, because I lead one of these teams, the one that is focused on cachexia, and the grant challenges are really a visionary way of funding that was initially conceived by Cancer Research UK, and that is now endorsed and co-supported by the National Cancer Institute and the National Institute of Health in the United States, as well as some of the cancer research foundations, the Mark Foundation for Cancer Research being one of them, for example. And so the idea here is that some questions are too big to be tackled by an individual or a single lab, and need a team effort that is interdisciplinary and that is enabled by less restricted funding so that people can engage in blue sky thinking and take on a question from a new perspective and perhaps find answers in places where we haven't previously been looking. And so these grand challenges have specific sort of topics that they address. And one of the topics that was funded now about 2 years ago is cancer cachexia. And David, Claire, and I are 3 representatives of this team. It's now got a team of almost 70 participants from all ranks of science, from research technicians to PhD students to postdocs to clinicians to professors and administrative support. So it's like a super lab between different groups that all bring their expertise to bear on this problem of cachexia. And we have team meetings regularly and conferences within our team where we exchange knowledge, and we are collaborating on research manuscripts that take on the questions in the way that none of us could take them on alone. So the work that we are delivering within this grand challenge is very different from the work that you would see from one isolated research lab. It transcends different research ideas and research approaches. And as a consequence, we are hopeful that we will make discoveries that we would otherwise not make in understanding how cachexia works and how we can treat it and how we can bring it to the patient.
Yeah, I mean, I think it's a unique and amazing opportunity to be involved in one of these grand challenge teams. And as we've been talking about in this call, cachexia is such a complicated subject with so many different systems at play that actually no single scientist has sufficient understanding of all of these systems in combination to really tackle the problem. And it's really at the heart of the full complexity of biology, and technology, really, to be able to develop these things. So it's fascinating. All of the meetings are very fascinating because you're always on the edge of your knowledge and you're learning a lot about other people's systems, whether that's looking at it from a neuroendocrine, like a brain hormonal system, or looking at different nutrients, different metabolism, or looking at the immune system. All of these things and all of these different models that we're using, as well as the clinical work, mean that I think it's a unique gathering of people to bring to bear on this challenge. And it certainly enriches our work to be able to feed directly into such a varied group of scientists, and it's been hugely stimulating. So I really hope it can continue and that this can be really the future of how we tackle these problems.
Yes, I think the CANCAN consortium has certainly provided an important platform for the sharing of interesting ideas and sharing of information and data sets. I think it puts us in a better position to be able to tackle the challenges of cachexia, as we've heard.
Thank you very much. So that brings us to the end of our discussion today. We've gained valuable insights into cancer-associated cachexia, thanks to our distinguished guest. Before we wrap up, I'd like to extend a heartfelt thank you to each of you for joining us and sharing your expertise today with us. And your works are crucial in enhancing our understanding of cachexia and also improving patients' lives. Thank you for all that you do. Thanks a lot.
Thank you.
Thank you for having us.